Hello and welcome to the Retina Radar podcast. I'm Arshad Khanani, Managing Partner and Director of Clinical Research and Director of Fellowship at Sierra Eye Associates and Clinical Professor of Surgery at the University of Nevada, Reno School of Medicine in Reno, Nevada. In each episode, I'll be sharing information on the latest breakthroughs, clinical insights, and practical pearls for your everyday use. We'll hear from leading experts, explore cutting-edge research, and discuss the trends shaping our field. Whether you're in clinic, in surgery, or on the move, Retina Radar keeps you informed, inspired, and ahead of the curve. To earn CME credit, visit mededtalks.com, search Retina Radar, or click the link in the notes to complete the activity and post-test. Before we begin, please review the faculty disclosures and learning objectives. If you have a question for our experts, send it to us at cme@vindicocme.com, and it may be featured in a future episode. This podcast is supported by an educational grant from 4D Molecular Therapeutics, AbbVie, Adverum Biotechnologies, Alkeus Pharmaceuticals, Astellas, EyePoint Pharmaceuticals, Genentech, a member of the Roche Group, Neurotech Pharmaceuticals, Ocular Therapeutix, Oculis, Regeneron Pharmaceuticals, and REGENXBIO. Today, I'm joined by my good friends and colleagues, Dr. Bob Avery, founder of California Retina Consultants, and Dr. Dante Pieramici, partner at California Retina Consultants. Both of you are legends in our field, right? You have been practicing for a while. You have seen pre-anti-VEGF days, and you guys were actually very involved in bringing new treatments for our patients. Really amazing job with research and leadership and podium presentation. And I'm so glad that both of you are here because you're always working together and innovating together. And it's a great synergy that I've seen. You know, some practices you have two big names, they clash. And here you guys are always cheerleading for each other and also for your younger partners like Dilshare and Nate. So really, I think we have a lot to learn from both of you about practice dynamics and how to work together and grow. But the main topic at hand today will be gene therapy for retinal diseases, and both of you are pioneers in that in terms of clinical trials and presentation and advising the companies. So before we get there, for our podcast, it's very common for us to kind of find out about each of you in terms of how do you end up doing what you're doing now and about your hobby. So Bob, we'll start with you. Tell us a little bit about how you ended up in retina and what's your favorite hobby?
Well, I guess retina is not necessarily a hobby, so you're right. I have another hobby, but I ended up in retina because of who I ran into in medical school. I had the privilege of running into a few great retina folks at Hopkins when I was in medical school with like Dante, but I ran into, I guess, Stuart Fine first pushed me in the field, but then I spent time with Bert Glaser and Ron Michaels, and it just seemed like a great field where you could do it all. You could be a clinician, a surgeon, and a researcher, and I really followed in their footsteps very, very minimally compared to what they did. But that's how I got into retina.
And what about your hobby? I know you like to sail, right?
Sailing. I race sailboats out here on wet Wednesdays, but the season just ended this Wednesday, so I have time for this now. But it ends with daylight savings time because it's too dark to race in the daylight saving time. But it's fun. It's something I figured I could do till I get really old. So I find it fun.
No, that's great. And so you work with two big names in the field, Ron Michelson and Bert Glaser, for sure. So Dante, you were with Bob in med school. Is that how it went?
Well, I'd like to think Bob was quite a few years older than me, but he was leaving medical school when I was coming in, and like a lot of people, I was sort of interested in neuroscience, neurology, neurosurgery. One of the neurosurgeons said, "Why don't you go and learn about the eyes? It'll be helpful." And so I had no intention of doing ophthalmology, but the Wilmer Eye Institute was a big deal, and you meet people there, and I think that I met people like Bob. Bob was my chief resident when I became a resident then at the Wilmer Eye Institute, and he was always, he was an easy guy to work with. He liked to play hard and work hard. And I thought that was a good combination. He'd take us out to the beach, the residents, but then beat us down with questions. No, I have a great picture doing my first cataract surgery with Bob. And if you look closely in the picture, you'll see I have two different gloves on because I contaminated one of my hands right away. I don't get that picture.
I taught you your first cataract surgery, which is obviously why you went into retina. I think that's probably.
Yeah, probably because I knew I would need to fix my own cataract surgery. But no, I think that Bob, I stayed on as faculty. Bob came out to California, and then I was renegotiating my salary. And Bob said, "Oh, just come out and use us as a bargaining chip." And so my wife and he brought us out in like January, and in Baltimore in January, snow drifts and cold, and Santa Barbara was like a beautiful day, sunny. I think the dolphins were jumping in the water, and we decided that we would stay. And doing clinical research turned out to be much easier in private practice, as you know, Arshad. I mean, in the academics is very difficult, takes so long. But in private practice, we are able to develop a clinical research center and really move on things. One day, we hope to have as busy of a clinical research project as you do, Arshad.
It's one of those things. You guys are doing great. I mean, you've done so many trials over the last 2 decades, and it's always fun to work with both of you. And one thing about Bob Dante, I'll say, he's just easy to work with. He just he's just a nice guy who was easy to work with. And
he's from Alabama. What do you expect? Right?
I have low expectations working here.
It's great. So Dante, what's your hobby other than growing long hair and be a model?
Yeah, well, I'm just trying to keep up with you, Arshad. I live in Santa Barbara. I rarely ever go to the beach. Unlike Bob, I like the other end of Santa Barbara, the mountains. I do a lot of cycling and running, trail running, and hiking and stuff. I like the mountains, so I spend a lot of time on the bike.
Wow. Great. Two legends with different hobbies. So I think whatever, in terms of like your relationship has always impressed me, as I said. So tell us, like, how do you guys work so well together? Like, I'm sure there are days when you're frustrated, but like you have built like an amazing practice where and I don't think I've seen a single practice like yours, Bob and Dante. Like you have like 4 or 5 people on the podium at every single meeting. You are making a difference. So what's the secret sauce in rising everybody together? What is the sauce?
I guess if I had to say what probably worked for me was I tried to hire people better than me. I wasn't, I didn't want to feel like, I would be intimidated by hiring someone better than me that would show me up. I figure my secret sauce is get the very best people you can get, and then you'll have an advantage over the other competition. And so that's what I set out to do. And
that's why we keep growing, because he's still trying to find someone better than him. So
exactly. So humble, right? But he's right. Like he's trying to get people everybody can learn from each other, right? That's the key. Like the day you think that you know better than others, then that's the day you stop learning. So I really like the philosophy, but it's a very simple secret sauce, it looks like, but it's hard to find. So no, congrats to both of you.
I'll throw one comment I learned sort of scrolling, unfortunately, on the phone because I'm susceptible. There was a comment I really liked. It said, if you're the smartest guy in the room, you're in the wrong room. So that's sort of what I applied to my practice. I just want people who are really exceptional and learn from them. Sort of like you said, Arshad.
No?
Yeah, and I guess the only comment I'd add to that is that, retina is a team sport. And I think that we've always had the attitude that we all work together, we share patients. It's not my practice in this office and someone's practice. And if we're a team effort when it comes to research, when it comes to patients. And our staff does so much for us, really. We've tried to find an excellent staff, and they really boost us up. And as you know, Arshad, when it comes to clinical trials, having good research people, an excellent research team, and good technicians and scribes really makes all the difference in the world.
No, 100%. I think that's a very valuable advice. A lot of pearls here from both of you already for our audience about building a great team of great doctors, doing research, being cutting edge, and at the end of the day, still being nice to colleagues. And I think that's the key here. Okay, so let's get to topic at hand. Gene therapy. We have had a lot of data recently over the last 5 to 10 years in terms of phase 1, phase 2 studies, reading out, and we have now 3 programs in phase 3 development. So I think let's get started with Cerevac or RGX-314. I know both of you are involved in the phase 1, and of course, Cerevac is being studied subretinally for neovascular AMD, and then suprachoroidally for neovascular AMD and diabetic retinopathy, which is quite exciting. But I think our focus will be subretinal for wet AMD. So Dante, I'll get started with you in terms of like surgical delivery. Like, a lot of people get nervous about putting something under the retina. And I'll tell you, who taught me how to do it well in a lab? It was Bob Abe. Bob Abe.
Oh, nice.
I was at a lab in San Diego, and he said, there are 2 ways to do it. This is how I do it. And guess what? It was very simple. I don't do touchdown and inject. I actually get close to it, and then I start injecting and then touchdown. And Bob, you probably don't remember because you were teaching a lot of other people, but that was like the pearl I got from you. And really, I've done so many of these without any difficulty. But Dante, tell us a little bit about, like, is it complicated? Is anybody can do it? What are the learnings from phase 1 in terms of positioning of the bleb and things like that?
Well, we had a little experience coming into this because we had previously been doing retinal translocations and, with the submacular surgery trial, doing SST type surgery, evacuating submacular hemorrhages with TPA. So I think we kind of honed in on those techniques where you would sort of, as you came down on the retina, you actually start, hydrodissect your way into the subretinal space. But yeah, I think the surgery is relatively straightforward. I mean, you do a vitrectomy and you use the subretinal needle and infuse and find a spot. We've over the years we've realized that you do get these RPE pigmentary changes, which is, it's not clear exactly what that is. Is it inflammatory? Is it the cell overactivated or just making too much of the protein and not enough of other stuff? But we've realized that we don't want to do this anywhere near the macula. We want to do this inferiorly. We make the bleb usually inferior nasally if you can. We sit the patient up after the surgery so that the drug itself will go inferiorly, because it doesn't need to be in the macula. When you're doing gene therapy like this, gene therapy is a biofactory. What we're trying to do is get cells inside the retina, particularly under the retina and in the retina itself, to start making this anti-VEGF agent, which, for Cerevac, it looks a lot like ranibizumab. So, by doing that, we do get some pigmentary changes, but the patients don't seem to complain about it. We can see them and they progress over a number of, probably 6 months they start and over a couple of years and then sort of taper off without any apparently visual significant complications if we position it in the right spot. So, yeah, I mean, I think that most surgeons really, if you're going to be a vitreoretinal surgeon, this is not one of the more difficult things to do, but I think it just needs to be done right. And I think that these changes that we've made from learning about these RP pigmentary changes, which they see in pretty much all types of subretinal gene therapy when you're using a viral vector, positioning it inferiorly, putting the patient up for the first 24 hours, really has made that complication or that apparent complication not significant for the vision.
No, I agree with you. And that's my experience. Of course, both of you are much ahead in terms of enrollment in the phase 1/2. And then I joined in, and obviously we learned a lot together. Bob, in terms of efficacy, I know you treated a lot of patients. The promise of subretinal gene therapy in patients with wet AMD, you've been in the space for a long time. Is it very exciting?
Absolutely. I mean, that study that we were in together, the phase 1/2, we ended up being top enrollers, but we didn't start till cohort 4 and 5, which were the higher doses and were the efficacious doses. I mean, the earlier cohorts, 1 and 2, really didn't do anything. 3 did some, but 4 and 5 worked the best. And it was really quite rewarding from having been injecting patients for so many years, to have a few patients that had one treatment and then never needed another injection. The true one and done that seemed to be so elusive. Unfortunately, as Dante mentioned, we first sort of saw those pigment changes in our patients and tried to, figure out what was going on. And we had somewhat intentionally placed the blood superiorly, thinking that this way you'd get more coverage of the drug as it spread through the macula and inferiorly. We didn't anticipate these pigment changes, and then we started seeing them in the far periphery of all places where you don't necessarily think your small blob is going to end up all the way up against the inferior aura, but that's where a lot of it went, because we sat them up and and there was an air bubble, and they're 8 years old, and it just moved. And so it was interesting to be part of figuring out what's going on. But as Dante said, it's not a problem now because we can put the blood inferiorly, and if there is pigmentary change, which there is in most of the patients, it's in the periphery, and it's like a sector PRP, if anything, which is a small price to pay for having high levels of anti-VEGF for years to come. And so it really was fun, though, to have patients that never needed another treatment. It was really exciting.
And you still have those patients, Bob? I would assume.
I still do. I still have them. They started old and they finished their 5-year long-term follow-up, and now they're back in my regular clinic. But, some have passed because of the ages, making it through COVID and all that sort of stuff. But and it's really a neat thing.
I just saw one today who is 2019. So yeah, I mean, these patients are out there, and it's neat to see these. And we see this with all the gene therapies that we've been part of. These patients that are out many years and doing well, and that's the amazing thing, really, is that the durability of some of these treatments.
Yeah, that's the point I was going to make, that I think people think, oh, how long is it going to last? And what I've seen in my experience too, like it's lasting for a really long time, 5 years plus. And then the other question people ask, well, you cannot reverse it. What if you want to stop the treatment? It's like, do we ever stop treatment for wet AMD? We really don't, right? Because we don't want to risk a bleed. So I think that's where gene therapy really brings value. And Dante, as you explained to the audience, that this is not gene replacement therapy or some missing gene. This is adding a gene. So using gene therapy as a drug delivery platform to carry a gene for a product like ranibizumab in Ceravec case, and then of course aflibercept in Exovac and 4D-150. But one thing I've seen with subretinal is that inflammation is essentially non-existent. Would you both agree with that?
Well, I think if you're talking about inflammation that you see in the vitreous cavity, yeah. I mean, it's not clear that some of the pigmentary changes potentially may have some inflammatory component to it. But I agree, you don't need to do prophylaxis with topical steroids or anything, or oral steroids for these cases. We've seen when you treat the fellow eye with Ceravec, you really don't get any inflammation, because it would be a real concern, I think, if you were developing inflammation and antibodies difference from the one eye treatment. At least from subretinal therapy, it looks like you can pretty safely treat the fellow eye, get the same efficacy and safety.
Yeah, which is actually very excellent data set. We are waiting for fellow eye data from other programs, obviously for intravitreal. So let's move on to intravitreal gene therapy. Let's start with 4D-150. Bob, what are your thoughts on 4D-150? This uses an R100 vector that takes a transgene for aflibercept as well as interference mRNA for VEGF-C, and this was studied in the PRISM phase 1/2 study, and the program is in phase 3 development in Forefront 1 and Forefront 2. What are your thoughts on intravitreal gene therapy with 4D-150 and your experience to date?
Well, it's another really exciting option, because they could be administered without going into the operating room. And I think that is of great appeal if you can get a similar sort of response. Now, I don't think that the intravitreal agents that we're using currently are quite as potent as, say, the optic trial where we had like tenfold what's being used by this other company. So I still think the subretinal may have the most potency, but we'll see. Time will tell as we look at it. But it's a great thing if we could just do an injection in the office and have anything like what we see with the cohort 4 and 5. The nice thing is that they're showing a marked reduction in the treatment burden, up to, I think, 83% reduction. And one interesting slide I've seen of theirs is that we have to think of when we look at all these studies about treatment burden reduction is that they have 4 different levels of treatment reduction burden depending on the severity of the patient enrolled in these, which are small studies. But people that are heavily dependent on injections have a lesser degree, maybe 44% of those guys are treatment free. And then, they still did well. They had a reduction. That's treatment free, mind you, not a reduction in burden. But it went up to 80% in the people who were just newly diagnosed and, didn't seem to have a high burden. And so we'll see this variability when we try to do cross-trial comparisons between the TKIs and all of these gene therapies. It's going to be hard to compare them because it really depends on how needy the patients are when they start the trial. The thing about the 4D is they also make a VEGF-C inhibitor. It's a small inhibitory RNA. VEGF-C isn't as validated as a target as it used to be before the AbbVie results came out, but I still think it probably helps because we see upregulation rather of VEGF-C when there's full blockage of VEGF-A and B, and so it may be a benefit. It's a nice thing to have there, but how important it is probably less likely to be critical now that we've seen the VEGF-C antibody results. But still, as an additive thing, that's probably useful. They had no significant inflammation, and I think it's viable. So we'll see.
Yeah, no, I agree with you. I think my experience is just like yours. I think safety is a priority, right, with intravitreal gene therapy where the vector is going everywhere. So the R100 vector is more retinotropic, and we're trying to avoid the front of the eye transfection, right? That's the key. And then lowering the dose, right? As you said, you can go very high and secrete very high level of proteins, but the problem is you pay the price on the safety side. And with all the great anti-VEGF injections available and tyrosine kinase inhibitor coming in, I think gene therapy, if it's going to be successful, has to be very, very safe. And the VEGF-C is interesting, right? We did not see superiority in the sozinibercept trial, but clearly, just like we didn't see superiority in Ang2 inhibition in co-formulation studied with nesvacumab and aflibercept, but a bispecific did help dry a little bit better. So the hope is that it may help a little bit, as you said. And what I also learned is that having that component increases the amount of protein that's being produced, just aflibercept. So it's very interesting dynamic, but yeah, very, very exciting. Dante, you've been involved with the program closely too. Anything to add on top of what Bob taught us?
No, I think you guys have touched the efficacy and safety. I think I actually got involved because I thought the bioengineering of this R100 was really fascinating. I mean, it's a chimeric vector. It's quite evolved through directed evolution in a primate model, and it has this dual transgene, which I thought was kind of cool. And so I said, "Wow, we should get involved with this," and we did. And as you said, I think that we're able to use pretty low doses of this and get a lot of efficacy. And it's probably in large part because of the R100 vector, and that transduces very well the retina cells. But it may be the payload too. The anti-RNA for the C may also help. We don't know, but we're using pretty low doses here, and going to be very low doses in patients who have diabetes that'll be investigated as well. So yeah, I think that to have an intravitreal injection that you only need to use topical steroids to prophylax over a period of time and really have very little inflammation, certainly no severe inflammation. We're able to get all the patients off this topical drop, and then it doesn't have a rebound. It's going to be pretty interesting for an in-office solution.
Yeah, no, I agree with you. I think the biggest thing for me from the program, obviously the efficacy, if you look at approximately anywhere from 50 to 80% injection free based on what kind of population you're looking at, as Bob said, I think chronic high need monthly patients are never going to be injection free because all you can produce is a certain level of anti-VEGF. And if they need more than that, they're going to need supplemental injections. So I think this idea of one and done, I don't think it's the right way to think about gene therapy, because it's a drug delivery platform and it's patient dependent because it's a heterogeneous patient population. So not all patients are going to be cured per se, but if more than half can be without injection and the others can have 70-80% reduction, I think it's going to be very well adopted. But as you both said, I think safety, obviously, it's a priority. So PRISM has shown very good safety, very comparable to anti-VEGF trials. But let's see what Forefront 1 and Forefront 2 show. We are going to find that out in the next few years. Let's talk about Spectra. I just want to let the audience know it was also studied in DME, as Dante said. And in that trial, actually, there was zero inflammation. So just showing that because of the low doses and the prophylaxis with topical, in Spectra it was 16-week, in Prism it was 20-week. I think patients have done really, really well. So excited to see DME program also moving forward in pivotal stages. Okay, let's go to XOAC, which is another gene therapy that uses 7M8 vector, takes a transgene of aflibercept. It's actually the longest data set we have in terms of intravitreal gene therapy, and really, kind of taught us a lot of lessons, right, from that program. As Bob, you were saying, the efficacy we saw in OPTIC with 6E11 dose was phenomenal.
That was amazing. 90%, just injection burden. And so many of those patients just injection free, period. And if you're injection free at 1 year, you are going to be injection free, except for maybe one patient. So it was fascinating. That's what really opened my eyes to the possibility of gene therapy and intravitreal gene therapy. But in the diabetes study, the Infinity, they did have some hypotony at that high dose, and it didn't respond to steroids as well. And so they've come back now with about a 10-fold reduction in the dose they're taking forward. And you still get very good efficacy. It's not the one and done as much as you get 10-fold higher, but you get rid of a lot of those potential side effects. And, with no one having to be on steroids at the end of the year, that's a big plus. I mean, you're balancing the efficacy versus the safety with these agents. And we see dose response curves in several of these different products, but you also see a dose response curve with the safety too. So the higher doses work better, but they have more safety concerns, whether it's episcleritis or whether it's intraocular inflammation and hypotony. And so how we use the steroids and how we use the right dose, it takes a little bit of trial and error, and that's why it's been the phase 2s have been really helpful in trying to sort this out for all companies. But I'm really happy to see this drug at tenfold lower dose doing so well, and I'm excited for it. I think it's definitely got a good shot, and it still has good PK. It's not as good as a tenfold higher dose, but that PK goes out for years, 5 years. I mean, it's amazing how much we, could benefit from these agents given intravitreally if we get the safety right with the right dose of steroids, in this population. But in general, I think it's a good balance between finding the right dose that's efficacious and finding it to be safe. And I think that's what they've done here.
Yeah, no, I agree with you. I think, as you said, we have learned a lot from this program, right? Like how to minimize the inflammation, the pigmentary changes, and how to balance that efficacy. Dante, you recently presented that data set from Luna. Any additional thoughts on it to summarize the data on top of what Bob already said?
I mean, the optic for neovascular AMD was definitely a very positive thing. I think the Infinity trial showed us really what can potentially happen and what we need to watch for. They sort of recollected their thoughts and they designed the Luna trial. So for two things, really, to answer the question, can we go to lower doses and still get efficacy, and which would put us at a more safe level? And so this is why they went to the lower doses in the Luna trial, tenfold lower, as Bob mentioned before. And the second thing was, what's the best way to prophylax these patients? Luckily, the second part that we learned was that topical steroids for around 20 weeks on a tapering schedule of difluprednate is very good prophylaxis, so very little inflammation. And the second thing we learned was that these lower doses were quite efficacious. They, 6E10 dose, and that this is a dose that you could take forward in a phase 3 clinical trial and have a lot of confidence that you're going to get good efficacy and good safety. And I think if you look at all these molecules, in the 4D-150, the Surovec, and the Ixovec, you're finding very similar results when it comes to reduction in the burden, when it comes to percentage of patients who are, injection-free, and safety is looking very similar between these. So I think, to me, that adds a lot of confidence that this gene therapy approach is good, because if one of them was working and the other two were failing miserably, I'd be suspicious. But since they're all doing pretty well, it adds a lot of confidence.
Yeah, no, I agree. I think that's a great summary, that the data of efficacy looks very similar. Safety, obviously, as we discussed, not much intraocular or none, essentially, from subretinal. Obviously, we use post-op steroids, but no prophylaxis. We have seen pigmentary changes under the retina. For intravitreal, lower dose, the better, and for D150, start with the lowest dose. Safety looks really good. Ixovec had that learning over the last 7-8 years, going from 6E11 to 2E11 to 6E10 now in the pivotal Artemis study, which is fully enrolled based on the recent press release, and their second pivotal should be starting in the near future. So really good summary for the audience. I'm sure all of us are going to learn in terms of how gene therapy pans out in efficacy and safety in the larger pivotal studies, right? First one, obviously, the data will come out from Surovec program, and then followed by these two programs. So I think next several years are very exciting. I know there's a lot of negativity in the community with gene therapy, and a lot of people feel like it's not the right fit, or they don't like the efficacy and safety profile. And between three of us, we have seen all these patients, right? And I think what is missing is that without seeing those patients who are really happy and controlled for such a long time after a single treatment, I think it's very hard to see the value. And, I gave a talk at Retina. I was invited to talk on gene therapy, and I said, whether you like it or not, it's going to be here. So I think that's the bottom line. I think one or two of these programs made the finish line, and it will be here. So I think it's good for all of us to learn about it and continue to learn, right? That's what we have been doing with gene therapy. So thank you for your time. I know we're running short on time, but we want to close this podcast, and we always ask this question to our faculty. So Dante, we'll first start with you. You've done a lot, published hundreds of papers, presented podium around the world. Be a part of a lot of recent developments in retina. What is something you're really proud of?
Well, I think I'm probably most proud of the California retina consultants that we've developed over the years. I mean, I've got to work with a bunch of great retina doctors, including Bob, who's, sort of led the way, and that we've taken this little practice over the years of the last 20, 30 years, and we've grown it. We've taken care of hundreds of thousands of patients and given them great care. We've been involved in hundreds of clinical trials. And, each of us makes a little teeny contribution to the science, but I think that our group has made these contributions and sort of helped push it a little bit forward. Maybe somebody else would have done it if we weren't there, but I think that it's been nice to be part of this team and to see this grow over the years. And now to be part of the bigger team, Retina Consultants of America, who we are one of the founding groups of that, I think really is what I'll take with me at the last as the most important thing that I've gotten out of my career so far.
Yeah, we should be very proud of what you have built with Bob. Bob?
I feel the same way. I think that it's been fun to come and develop, a really stellar retina group in an area that didn't have that great of retina care, because the whole Central Coast here was, it wasn't that well supplied back in the day. But I really have enjoyed being part of that. I've enjoyed also being a part of the VEGF sort of discovery back when I was a chief resident, and we sort of helped. And most of the cases in the New England Journal paper identifying VEGF and retinal disease that we published with Lloyd and Napo, most of those cases came from Santa Barbara because I started it when I was there and I brought the project here, and that was fun. And then being the second group to start injecting Avastin, in humans after Phil sort of gave me the nod, and then ending up in gene therapy, getting VEGF into people's eyes via a vector. It's been a 30-year trek from start to finish, but it's sort of a nice bookends to it. And doing it only because I have great partners at our practice, as Dante said, that have really taken the ball and run with it. So it's been a good run, and it's been fun to work with everyone, and appreciate you inviting us to be part of your podcast, Arshad. Thank you.
No, you guys are
been great.
You guys are both excellent. So before we go, Dante, give me like 10 seconds on how you would remember Bob when he retires.
The thing I've loved about Bob, and it's probably a lot of the things I loved about working with people like Jean Dewan, is that, there's never really the impossible. It's like I say, well, we should try to do this. Let's do translocation. Let's do 25 gauge surgery. Let's do anti-VEGF injections, even though there's, only 2 or 3 patients that have been done in the country. Bob was always game for that. And I think that when you've got the senior guys opening the door saying, do whatever you want, and let's get involved, and let's be creative, and let's be innovative, I think that that's been a good inspiration to me.
How would you remember Dante?
Well, Dante was the top surgeon at Wilmer when he was plucked when I stole him. But not only has he brought the surgical level up in this community, but he's also run our research department. And you, Arshad, know what's involved in that. He has built it from a small little side thought into, maybe not quite as big as yours, but a big research.
One day.
One day, we're all aspiring to be like. We don't see how you can possibly get so many patients so fast. You're amazing. So we got someone to aspire to copy. So we're getting there. So, but Dante has been in charge of that. Let me deal with -25s and boring stuff while he does the exciting running the research department. I of course like to participate, but he gets kudos for doing that.
You both are amazing. It's so great to learn from you and speak to you. We could have gone hours about, your learnings from each other and learnings from the field. And Bob is just a phenomenal human being. And I know you have trained so many. And our good friend, Carlos Gazara, was also trained by you. And he's a great friend of mine and really an amazing person to work with. So thank you, Bob. Thank you, Dante, for being on this podcast. I also want to thank the audience for listening to it. Thank you both. Have a good one.
Thanks, Arshad. It was a lot of fun.
Thank you.
To earn CME credit, visit mededtalks.com, search Retina Radar, or click the link in the notes to complete the activity and post-test. Have a question for our experts? Send it to us at cme@vindicocme.com, and it may be featured in a future episode. Stay tuned for future episodes in the Retina Radar series to keep up with the latest insights and expert discussions.